Well, hello again. So for the past few weeks, we've been talking about uh, molecules and how they are metabolized, either constructed or broken down to release energy. Today we're going to shift gears a little bit, and not going to talk about any individual molecules, but we're going to look at some basic principles of genetics. I'm sorry about getting this out to you a little late. I switched computers and had little technical difficulties, so I've had to switch to actually a third computer, but that one seems to be working. So let's take a look at what we have here. So back in the mid 1800s, this man, Mendel, was maybe not the first person to ever do any kind of genetic experiment, but certainly the first person to really uh, systematically look at it and write it down. And that's very important. If you don't write it down, then it's it's like you haven't done it. But he did a series of experiments that really laid the foundation of genetics for the next um, 175 years. Um, Now, some of the things he did might be a little questionable. And of course, back then, things were, the scientific method wasn't quite as rigorous as it is today. So we can maybe forgive a few little, taking a few little um, liberties. Um, but let's look at the definition to start with. And genetics is simply the study of heredity. There's some modifications here. You can look at them if you want. But basically what we're looking at is how different traits are passed on from one generation to the next. Okay, we're going to talk about some basic concepts in genetics here first, and then talk about mono and dihybrid crosses, look at what Mendel concluded from his experiments, and then we'll take a little look at how modern understanding of Mendelian genetics works. But before we go on, I just want to say I uploaded this lecture yesterday, got an email this morning saying that the sound cut out after just a few slides, and so I'm redoing that as I speak. But please, in the future, if this happens or if there are any other problems, email me as soon as possible so we can correct that. Okay, so let's look at a monohybrid cross, and that is now we're just following one trait. So in this case, in Mendel's case, he was working with peas, and he had two lineages of peas, one that would produce tall plants and one that produced shorter, what he called dwarf plants. So he took a tall plant and mated it or crossed it with a dwarf plant. And what he found in what we call the F1 generation, that's the first generation, was all tall plants. So every one of the progeny was a tall plant. Okay, when he took the F1 generation and crossed them to each other, so mating siblings, and we can do that with peas, but we don't generally do that with humans. This is what he got. He got for every 4 plants, there were 3 tall ones and 1 dwarf one. Now in the F1 generation, every plant that he looked at would be tall. With the F2 generation, this is the ratio that you get. So it's not that every 4 plants have this specific ratio. It's overall the ratio will be 3 to 1. Okay, so from this, and I assume a lot of thinking, Mendel came up with these ideas. And what he called unit factors, which we now call genes, are present in pairs. And these genetic characteristics are controlled by these unit factors. And again, each one of these exists as a pair in each individual organism, or now we know in individual cells. If there are two genes or the factors that carry out the same function that are different from each other, that behave differently, and generally one is dominant over the other, so we call that a dominant version of the gene, and one is recessive. Okay, the third thing he noticed was that during the formation of gametes, the germline, the paired unit factors separate or segregate randomly so that each gamete receives one or the other with equal likelihood. And that's pretty much the case, although there can be some slight variation in that, but it's more likely due to the gametes being more robust than with one version than the other, than that they actually don't randomly assort. Okay, so let's look at the gene level. Up in the left-hand corner, we're starting with the two parents, the tall plant, which is the two genes from that plant are designated with capital Ds, and the dwarf plant, where the two genes are designated with small ds. And when you mate those, then the F1 generation gets one factor from each, so it's going to end up having one tall and one dwarf factor. And in this case, the tall version is the dominant one, so if you have one capital D or tall gene, you're going to have a tall plant. So all of the progeny from this are tall. Now when you call sibmate those, so you mate those to their siblings, and now each one of those F1 plants had one tall gene and one dwarf gene. So now when you mate them, you get a combination of different makeups shown at the bottom. So you can get a quarter of your progeny are going to have two tall genes. They got a tall gene from each of the parents, the F1 parents. Half of them will have one of each, because they got one of each from the parents. Half of those got the tall one from the male, half of them got it from the female. And then a quarter will have two dwarf genes, so it will have the dwarf phenotype. So it'll have three tall phenotypes, the one with two tall genes and the one with one each, the two with one each, and then one dwarf gene. And we can see this a different way in what's called Punnett squares, where we put the genotypes of the two parents, the male across the top, the female down the side, and then we fill in the boxes based on what those genes are. And again, we see the same thing in the F1 generation on the left. All four squares have one large D, one small d, and since they have a large D, they will be a tall plant. When we go to the F2 generation, and now we've mated two plants that have one of each of these types of genes, and so you get this configuration. Now, one thing about Mendel's work, we don't know whether he was lucky and the tall plants that he used had two tall genes in them, or whether he had a couple of trial runs where he couldn't figure out what was going on because it actually, the tall plants actually had one of each. But anyway, he managed to figure out what was going on at a time when really we didn't know any genes were unknown, chromosomes were unknown. We didn't know what the molecule that stored this genetic information was. Okay, I'd like to go over some terminology now so it would make it easier to talk about this genetics. And of course, a gene is a sequence of DNA of bases along the DNA that codes for a protein, originally called a unit factor by Mendel. But a genetic locus is a specific position or location on the chromosome, and really those, for the most part, those two terms, gene and locus, are interchangeable. So when we talk about a locus, we're referring to a specific gene. Now, each loci or gene can have variants, and those variants are called alleles. They're alternative forms of the gene. And we just saw an example of that with the peas, where we saw two variants of the gene that coded for the height of the plant. One allele or variant conferred tallness on the plant. The other was for a dwarf or a short plant. And some genes can have many different alleles that can have slightly varied expression. Now, the other thing we want to talk about is the idea of a homozygous organism, and then a diploid organism where all the cells have two chromosomes. If both of those alleles are the same or identical, then they're homozygous. If they're different, then they're called heterozygous. In the example of the peas, the parent and the crosses, the parent strain, the two parent strains, one of which was tall and one was dwarf, were both homozygous. They both had two identical alleles, two tall alleles and two dwarf alleles. In the F1 generation, all of those were heterozygous. They had one tall allele and one dwarf allele, getting one tall from one parent and one dwarf from the next parent. In the F2 generation, there were a mixture of homozygous and heterozygous plants. Okay, we're going to complicate things a little bit now and talk about two traits at the same time, or a dihybrid cross. In this case, we're still looking at Mendel's peas here, and so we're looking at the actual pea or the seed. And one trait is for color, either yellow or green. The other is for the texture, which is either smooth or wrinkled. So again, Mendel took two peas, one that was yellow and smooth, and crossed that with a green wrinkled pea, and all of the F1 progeny were yellow and smooth, which indicated that yellow and smooth were dominant over green and wrinkled. Now, once again, it would have been quite a stroke of luck for Mendel to have stumbled on two homozygous plants for both of these loci. And so it's likely that he looked through a couple of different scenarios until he got one that made some sense to him. At any rate, since yellow and smooth are are dominant, all the F1 generation are going to be yellow and smooth, but if as what happened here, the original parent strain were both homozygous, then all the F1 strain progeny are going to be heterozygous with one yellow, one green allele, and one smooth and one wrinkled allele. Now when you take those and cross them back to each other, and again they're all identical, so it doesn't matter which ones you use, you get these four variants shown on the right here in the F2 generation, and the ratios given here. So 9 of the 16 are going to be yellow and smooth. Now remember, you only need one yellow to get one yellow allele to get a yellow P, and one smooth allele to get a smooth P. So 3 of the 16 are going to be yellow and wrinkled, and 3 of the 16 are going to be green and smooth, and just 1 out of 16 will be green and wrinkled. And if we look at the next slide, we can see why that is. So this again is a Punnett square, but showing all 4 possibilities now that include 2 alleles from 2 different loci. And so these are all the 16 different combinations, and you can see, remember once again, if there's a smooth, if one of the alleles is smooth, it will be smooth. If one of the alleles is yellow, it will be yellow. And so 9 of the 16 possibilities will give you a smooth yellow P. 3 of the possibilities will give you the recessive wrinkle phenotype, but still the dominant yellow one, and then 3 others will give you the opposite, the recessive green with the dominant smooth phenotype. And just 1 out of 16 now will acquire both dominant alleles for both loci. So remember now, if you look at just 16, you're likely not to find exactly these ratios, but if you look at enough of them, they will fall into these ratios. It's like gender in humans, which should be random, so half the progeny should be male and half should be female, but that doesn't mean every family has 1 of each or 2 of each or whatever. It means if you look at a large population, pretty much half will be female and half will be male, and so that's the same thing that's going on here. So let's review the postulates of Mendel now. Again, there are unit factors, which we now call genes, that of course he would have no idea of knowing. They coded for proteins, but are responsible for expressing certain traits, and these are found in pairs in most organisms. Often the 2 pairs are different, slightly different, have different properties, and generally one is dominant over the other one being recessive. During the formation of gametes, the paired unit factors or genes separate or segregate randomly. So each gamete receives one or the other of the 2 alleles with equal likelihood. And then those are assorted independently from each other. Okay, so this picture shows a picture of replicated chromosomes. So we have 2 chromatids, often called sister chromatids, that are essentially identical to each other. So if we look at these during meiosis, again starting on the left, we've got a duplex organism. So it has 2 homologous chromosomes, or a pair of each chromosome. Um, and this is showing one case where there are two different alleles. After replication, we get the two sister chromatids. So they're still attached at the centromere. Then those divide into two. So each sister chromatid goes, or the two homologous sister chromatids go in separate directions. And we get a second division which separates the sister chromatids. So now we've got four haploid cells. Now in most organisms, only one of those, or not all four of them anyway, are going to become germ cells for the next generation. Some very simple organisms like yeast, all four of the haploid cells will be used to produce the next generation. That's one reason why a lot of genetic study has been done in yeast, because you can follow each of the haploid cells after meiosis, whereas in other organisms like humans or flies or frogs, you can't do that. You can only see the result of one of them. Okay, a few more definitions. We've already used these words, but let's make sure we understand them. Diploid are cells that contain two sets of chromosomes, and that's most cells in our bodies. Haploid cells are cells that contain just one of each chromosome, and so basically those are gametes, egg and sperms. Now there are some cells in humans that are polyploid, meaning there are more than two copies of each chromosome and multiple copies of each homolog. So there are different, multiple different chromosomes. There are still two, but they've been replicated, but not. But the cell hasn't divided. In some other organisms, like insects, a lot of the tissues, particularly in the larva, have many copies of each chromosome, in some cases thousands. And so they're giant cells with lots of DNA. Okay, the other two definitions is, and we want to contrast these, the difference between phenotype and genotype. The phenotype is the observable properties of the organism, whereas the genotype is the specific allele, or the genetic constitution, or by today's standards, the DNA sequence. Now you can have alleles that have different genotypes, in other words, have variants in the DNA sequence, but have the same phenotype, so are still expressed the same way. Now, as we've seen, if two different loci are on different chromosomes, they will assort independently. So if we start with a chromosome or a gamete with a yellow allele on one chromosome, a white allele on the other, and the other gamete has a green and a black, we can end up with either yellow and white or yellow and black. Okay. Now, if they're on the same chromosome, of course they're going to sort together, what we refer to as they're linked. Although that's not always the case. So if they're close together, then they will almost always, if not always, pretty much always assort together. If they're farther apart on the chromosome, due to chromosomal rearrangements, which we call recombination or crossing over, they won't always assort together. So the farther away they are, the more often they won't assort together. And if they're at opposite ends of the chromosome, there's often enough recombining that they will appear to be on different chromosomes, or almost appear to be on different chromosomes. You can use that phenomena to map genes along the chromosome, particularly if you have a marker that you know where it is. You can determine the order of genes along the chromosome by how often they assort together or how closely linked they are. Okay, so again, questions that Mendel answered were the idea that there are these unit factors, or what we call genes now, and there are two of each one in each organism, and now we know in each cell. If those genes are different, we call different alleles, often, or more often than not, one is dominant over the other one, which is recessive. So if you have one copy of the dominant gene, you'll get that phenotype from that gene. If you have two copies of the recessive gene, then you'll have the recessive phenotype. There's the units separate or segregate randomly so that each of the gametes receives one or the other with equal likelihood. And during formation, there's independent assortment of the different unit factors or genes. And of course, that is if they are on different chromosomes. If they're on the same chromosome, now we know that that's not the case. Now, knowing what we know today, we can relate that to what's now called the central dogma of molecular biology or molecular genetics, where we have a DNA that stores genetic information in the form of different genes. Those genes can be transcribed into an RNA molecule, which then can be used to translate that information into a protein. And one gene can affect more than one trait, so it can have a pleiotropic effect. And any trait can be affected by more than one gene, and that's probably more often the case. And just a simple example from, again, from fruit flies, there are two pigments that are involved in the eye color of the fly, as well as other genes that are important, including allowing the deposition of the pigment, and so that any one of those genes can affect the different eye color. So, and many traits are affected by environmental factors as well as genes, and so you may have a situation, particularly during pregnancy, where something affects the fetus, causing a change in expression at critical moments during development, and that can have an effect on the developing fetus. Okay, now since Mendel's time, there's been a lot of progress made in genetics, and Mendel laid a very simple foundation, but there are a lot more details and interesting issues, and here's a list of some of the genetic issues that we're going to talk about for the rest of this lecture. The idea of incomplete or partial dominance, codominance, haploinsufficient, polymorphism, lethal alleles, gene interactions, sex linkage, the gain or loss of function mutations, and finally crossing over. Okay, so we talked about situations where you have one allele that's dominant over the other, like the tall plants are dominant over the dwarf plants, the yellow colored pea is dominant over the green pea, as well as the smooth pea is dominant over the wrinkled pea. But that's not always the case. Sometimes we get something in between, and that's sometimes called incomplete dominance. So in this example of flower color, we mate a red flower with a white flower, and the F1 progeny are pink. Okay, so in this case, the red was homozygous for the red allele. The white was homozygous, in other words, had two copies of the white allele. So every one of the F1s acquired one red allele and one white allele. The result was a pink flower. Now, when we cross the siblings from that F1 generation, we get a mixture shown at the bottom left. So we get one red for every two pink for every one white. And if we look at the Punnett square, you can see what's going on here. So in the F1 genotype, we've got one red allele, one white allele, the same with for both the female and the male. So when we cross them, we get every combination. And this results, again, in the ratio of one red to two pink to one white. And again, if we look at any four flowers, they're not necessarily going to have that ratio, but when we look at a large number of them, that's the ratio, or very close to that ratio, that we'll find. Now, the idea of haplosufficient and haploinsufficient means that in a haplosufficient gene, that means that a single copy is enough to make for normal function. In a haploinsufficient loci, one copy is not enough. You need two good copies in order to get the normal function. One example, and I keep coming back to the fruit fly Drosophila because that's where my background is in the what's called the indirect flight muscle in the fruit fly, and that's this. It's the largest muscle in the fly, and it's involved in the flight of the fly. And if you have only one actin gene, which of course actin is one of the major proteins in muscle, it's not enough to make enough actin for the fly to be able to fly. So this is an example of a haploinsufficient loci. If you have two good copies of the actin gene, it will make enough actin protein in the indirect flight muscle so the fly has normal flight. But if there's only one good copy of the actin gene, then the fly cannot make enough actin to make a viable flight muscle, and so the fly won't be able to fly properly. People that work on the indirect flight muscle, they test these flies by taking a large graduated cylinder and coating the inside of it with vacuum grease. And then they dump the flies into the cylinder and see where they end up on the side of the where they light on the side of the cylinder and stick into the grease. And so those that can't fly very well fall to the bottom. Those that are able to fly, you find closer to the top. Now, codominant is a situation where you have um, two alleles that produce different products that are distinct and detectable. Um, so in the case shown on the next slide, we can have these blood antigens or red blood cell antigens that are recognized by different antibodies. So we can have an A antigen or a B antigen, or in fact, no antigen. So different people have type A blood, uh, some people have type B blood, and some have type AB. So they have both the A antigen and B antigen. So two different alleles. So those people are heterozygous, or they could have neither antigen. This shows the makeup of the different uh, carbohydrates on the cell surface that react to different antibodies, so make up these different antigens. So these different blood types are found in the population, and so what we refer to those as polymorphisms. So these are different alleles that give a different phenotype. In this case, they're all considered normal, um, but they are different and distinct. Um, at the molecular level, we call a polymorphism anytime there's a base change, and often those base changes don't end up uh, changing the protein to any distinguishable amount. In fact, many of them don't change the amino acid structure at all, but at the DNA level, they're still polymorphisms, and we can use those to identify different alleles. So you can create novel phenotypes by mixing and matching these different alleles, and this is an example of all these different squashes that are shaped by different alleles, again, of the same loci. And so two loci can interact with different alleles to make a wide variety of shapes and sizes that we saw in the previous picture. Now you can get lethal alleles that are carried along in the population as a heterozygote. And there's an example of one of those. The normal mouse shown on the left is got hair or fur color that's called a goody. It's kind of a brownish color. It's got I think, a dark tip on the end of each hair, and then it's actually different colors along the length of the hair. And there's a mouse on the right that's yellow colored. Now, in this case, if you mate two agouti mice, then all the progeny are agouti. If you mate two yellow mice, 2/3 of the progeny are yellow and 1/3 is agouti. And if you mate a agouti with a yellow mouse, then it's half and half. So what's going on here? That's shown in these series of crosses. On the left, the two agouti mice, which turns out agouti is recessive to yellow, um, only make agouti offspring because they contribute two agouti genes to each of the F1 generation. If you mate two yellow mice together, now yellow is dominant, but it's also a lethal uh, allele. So all the yellow mice that you see are heterozygotes, having one yellow gene and one agouti. And if you have that uh, that set of alleles, you're going to be yellow. But when you cross them, if the F1 generation gets two agouti alleles, of course it'll be agouti. If it gets one of each, it'll be yellow again. But if it gets two yellow alleles, it'll be lethal. So, and you'll never see that mouse. So the ratio of offspring will be 2 yellow to 1 agouti. If you cross an agouti mouse with a yellow mouse, again, the yellow mouse has to be a heterozygote because a homozygote doesn't exist. It'll be lethal. Then half of the, let's put it this way, each of the F1 generation will acquire an agouti allele from the agouti mouse, and then half of them will acquire a yellow allele and half will acquire an agouti allele. So you'll get a half and half ratio. If the loci is on the X chromosome, the genetics is a little bit different in most organisms. The female has two X's and the male has only one and has a Y chromosome. I think in birds it's actually the other way around, but most organisms are this way, and that includes insects like the fruit fly. And so this is an example of a X-linked gene that we call white. And in Drosophila, the gene is named after the mutation. So if you knock out that gene and you get a white-eyed fly, then that is called the white gene, even though that gene is actually involved in normal red pigmentation. It's actually involved in the deposition of the pigmentation in the eye. It doesn't make the pigment. So if you have a white, or excuse me, a wild type or red-eyed female, and you cross that to a white male, what you'll get is all of the male progeny will be red-eyed, normal colored eye, wild type eye, because they will have inherited the X chromosome from their mother. All of the female flies will be heterozygotes because they will have inherited the white gene from the single X chromosome from the male. So keep in mind here that all males get their X chromosome from their mother. Now, if we do the opposite cross, white-eyed female to a red-eyed male, and again, there's only one allele in the male because it's on the X chromosome, and so everybody, all the females will get the red allele from the male, so they will have red eyes. And then all the males will get one of the white alleles from the mother, and so it'll have white eyes. So if you cross a homozygous white red-eyed fly to a red-eyed male, then you'll get the opposite. All the females will have red eyes, all the males will have white eyes. Okay, so we can have gain of function mutations as well as loss of function mutations. And the loss of function type of mutation is pretty straightforward. You know, if you have a amino acid base change that causes an amino acid change, that can destroy the protein, and so that would be a loss of function. Also, if there's a deletion of the gene or part of it, you could lose function. Gain of function mutations are rarer, as you might imagine, since what we're talking about is a change that produces a new function. And you can, one example might be if you have a receptor protein and a change in the region of the protein that identifies the ligand now binds to a different ligand or a slightly modified one. So that would be a gain of function. If it wasn't for gain of function mutations, well, we wouldn't be evolving at all, organisms in general. We would only lose activities rather than gain them. Can also have gain of functions that are mixing of or DNA rearrangements that mix different parts of different genes to form new functions. And I'll show you one in a few weeks where what's happened is the control region on one gene has been juxtaposed with the coding region on another, causing that gene then to be expressed in a different tissue, and it has pretty dramatic results. Okay, as I mentioned before, during mitosis or meiosis, we get, after the chromosomes are duplicated, we get what's called crossing over or homologous recombination. And these are exchanges of DNA between homologous chromosomes. So there's no gain or loss of function here. And we're going to talk about this in more detail later. But again, as shown in this schematic, if the two genes are close to each other on the same chromosome, then it's unlikely that a crossover event will be between them, and so they will tend to assort together through meiosis. If they are at farther apart or at extreme ends of the chromosomes, then the likelihood of a crossover event being between the two genes is greater, and so those two genes will often assort independently. If on a fairly large chromosome, if they're at the extreme ends of the chromosome, it's almost like they're not linked to the same chromosome. Okay, while Mendel did a lot of the groundwork and the basic, straightforward genetics, at the beginning of the 20th century, there were more geneticists. This is one, Thomas Hunt Morgan, who really pushed the field a lot farther. And rather than working with plants, Morgan worked with Drosophila, or the fruit fly. And again, he understood the idea of this crossing over, and you have to remember that at this time people didn't understand the concept of a chromosome or that DNA was what was storing the genetic information, but he realized that genes could be linked together, and sometimes they were very tightly linked and sometimes they were very loosely linked together. And I think I mentioned before, you can map the genes along the chromosome based on how closely they are linked to each other. So you measure the frequency of recombination between two genes, and that will tell you how far apart they are. Well, you have to realize that the genetic map does not, although it's in the same order as the physical or molecular map, it's not on the same scale because recombination occurs more frequently at the ends of the chromosomes or the farther you get from a centromere. And so those genes out there appear to be farther apart than the ones closer to the centromere. All right, we can use genetics to construct pedigrees in humans and track genetic diseases. And a couple of symbols here. When we draw these pedigrees, we use a symbol of a square for the male, circle for the female. If we're looking at a particular trait or disease, those that have that are the square or the circle will be filled in. A line drawn between a square and a circle indicates that those people are married. A dashed line indicates that they have, it says extramarital sex. I think it would go a little farther because it would mean that they're not married but have produced offspring, and because whether they had sex or not is irrelevant. If they have produced offspring but were married and now divorced, there'll be a slash line through. So in many cases, one of the parents, male or female, could have produced offspring with more than one partner. So a simple pedigree might look like this. In this case, you have the male and female parents. They produce 6 offspring. 2 of them have the issue, whatever that might be. And you can begin to tell even in the F1 generation whether it's an X-linked mutation or variant, or whether it's dominant or recessive. And it's easier to tell in one generation if it's X-linked, if you have enough progeny. It might take you a couple generations to tell if it's dominant. You can try to sit down and figure that out if you want. Now, one trait that's fairly common in the population is color blindness, and it only affects males. I should say primarily affects males at a vastly higher rate. So we should be able to determine if that's on the sex-linked chromosome or an autosomal chromosome, and whether it's dominant or recessive. And so if we have a recessive X-linked loci, and that's what color blindness is, can the son be unaffected, or son of unaffected parents be colorblind? And of course the answer to that is yes, because if the mother is a heterozygote for color blindness, the son will receive its only chromosome from its mother, and so it has a 50-50 chance of inheriting that affected chromosome. So again, this is a recessive single loci that's located on the X chromosome, and about 1 in 4 I think males are infected. Now there's one last concept that I want to talk about, and that's the idea of penetrance. And this is an example of a disease, Huntington's disease, which affected a a very well-known folk singer, Woody Guthrie, who died from Huntington's disease. He has also a very well-known son who's a folk singer, Arlo Guthrie, who appears not to have the disease. The picture on the right is of Arlo. He's probably in his 70s these days, so that was taken quite some time ago. But Huntington's, and let's look at the pedigree on the next slide. And Huntington's is actually caused by a series of 3 base pair repeats. And if you have the wrong number of them, then you can have this neurodegenerative disease that affects people in middle age. It starts to affect them in their 30s or 40s. And depending on how many repeats you have, it may be more severe than not. So there's a range of repeats where it's pretty much everybody is going to be affected. There's an intermediate range where it's hit or miss whether you have the disease, and of course if you have the right number of repeats, you'll be healthy. So this is an example of varying penetrance here. So not everybody with the genetic lesion has the disease, and that's often the case with genetic diseases, that they're not always 100% penetrant, meaning not everybody is affected that has the genotype. So if 60% of those that have that genotype are affected by the disease, then we would say there's 60% penetrance. Okay, so those are some basic concepts of genetics. Went through them pretty quickly. Most of you have probably seen most of those, if not all of them, before. So next time we're going to get back to metabolism and talk about one more group of molecules and how they're made or degraded. And I believe that will be the last group of molecules that we look at, their metabolic pathways.